Ladies and gentlemen, I'm Arthur Chen, and thank you for your kind introduction. The title of my talk is the glycine amyltransferase, a promising drug screening approach for the treatment of hepatocellular carcinoma. Okay, so I will give a short introduction of this molecule, this protein, or we call it enzyme, glycine amyltransferase. We call it GNMT. GMT is very abundant in the normal livers. It was estimated is about 1 to 3% of the total cytosolic protein. And GMT regulate the ratio of adenosine methionine to the S-adenosine homocysteine. That's the same as SH. So it participates in this pathway. So basically, GMT will grab the methyl group from SAM and transfer it to glycine. So the glycine will be methylated to become sarcosine. And then the SAM will become SAH. And then SH will be further metabolized to homocysteine, and then through this one carbon metabolism pathway. And because GMT is a major folate binding protein, and its enzyme activity will be inhibited by 5-methyl tetrahydrofolate. So it also played a role in the DNA synthesis, besides the regulator epigenetic modification of DNA. And we are the first group in the world identified GMT was downregulated in hepatocellular carcinoma, and that's in 1998. And then we also, so this is the immunohistochemical staining of liver tissue, and you can see that the GMT was downregulated in both tumor nodules, but it's still expressed quite abundantly in the tumor-adjacent normal livers. And also when later we used the qPCR to compare the expression level of GMT in hepatocellular carcinoma tissues. That's a T, and then it's and compare it's the expression in the tumor-adjacent tissue in HCC patient without any hepatitis infection, or 51 HCC patient with HBV infection, or 57 HCC patient with HCV antibodies. But in these three groups, you can see that GMT expression level was significantly downregulated in the tumor tissue compared to its tumor-adjacent tissue. Besides that, in 2003, in collaboration with Dr. Kenneth Butel at the NCI, we identified the genotypes of GMT, including some short tandem repeat polymorphism and the SNPs. And we found that GMT locus was 40% has loss of heterozygosity in HCC patient. If you need more information, I will encourage you to have this to read this review articles just published recently by the Italian group. That the title of this review is GMT is a multifaceted suppressor of hepatocarcinogenesis. So the first clue that we think GMT is a protector for liver cancer tumorigenesis is the paper that we published in Cancer Research. Here we shows that if we express the GMT at a different level using the adenovirus as a construct, as a vector, then we treat the cells, liver cancer cell lines, with benzopyrene. We know that benzopyrene is an environmental carcinogen. Then you can see when the cell express GMT, the level when it increase, then the DNA adults formed by this benzopyrene will decrease. Another experiment is we also found that GMT can also protect the cells from insult of the alpha toxin B1. As we know that alpha toxin B1 is also food contaminants that can also trigger tumorigenesis in liver. So in 2013, we published a review article in Toxicology Applied Pharmacology. In this review, we shows that the function of GMT in the xenobiotic detoxification pathway is when GMT expressed abundantly in cytosol, which is in the tetramer form, when the benzopyrene or alpha-toxin B1 carcinogens enter the liver cells, it will enhance its detoxification pathway and convert all this and carcinogen to non-genotoxic metabolites, and then pump it out through this phase 2 and phase 3 enzyme. But if there is no GMT present, for example, in liver cirrhosis patient, GMT was downregulated, then this carcinogen enter the cells and they will enter the nucleus and form the DNA adults. And all these genotoxic DNA adults will cause the mutation and tumorigenesis. Another evidence showing GMT has a protective role for the carcinogen insults is we generate a GMT transgenic mouse model. And here we use PPCK promoter to drive the GMT cDNA and overexpress this in the mouse liver. And then we challenge the mice with alpha-toxin B1. As you can see here, compared to the wild type, that 67% of the wild type mouse generate liver tumor. All the GMT transgenic mice do not have liver cancer. So GMT has a protective role against the carcinogen insults. Another interesting observation is, besides its enzymatic function or as a carcinogen binding protein, in 2012, we published a paper in Molecular Medicine to show that we use YEAST2 hybrid system to identify protein interacting with GMT, and we found this interesting protein called deptol interact with the GMT, and deptol is an inhibitor of mTOR. So, and we also found that in the HCC cohort in patients, we found that for patients whose tumor cells express high level of deptol than the tumor adjacent cell like this one. And you can see that its 5-year survival is very, very poor compared to another group of 37 HCC patients whose deptol expression in the tumor cell is less abundant, less obvious than the tumor adjacent tissue. Its 5-year survival is more than 65%. So higher expression levels of deptol is significantly associated with poor prognosis, especially in the HBV-related HCC patients. In addition, we produce a GMT mutant form because previously people found that there's an inherited congenital deficiency of GMT there's a point mutation at 140 amino acid of GMT. And this mutant form of GMT will lost 95% of its enzymatic activity, but its a 3-dimensional structure remained. It's quite similar to the wild type GMT. So we use the typical functional assay in the Q7 cell, and you can see that this N140S mutant here, this one, okay, of GMT still possess almost identical and equal function as the wild type GMT in counteract with this deptol function of inhibition of mTOR. So here we further shows that in the cells, overexpress of GMT compared to the GFP control, its cell survivals was decreased. And also it has a G2M phase progression, delayed progression, G2M phase delayed progression. So because of this, it will cause the senescence of the cells, and due to other factors, all this factor will cause the senescence. But if we treat the cell together with rapamycin, then it will cause the cell death. Another molecule we think is very interesting interacting with GMT is this PREX2. PREX2 has been found as an inhibitor of PTEN. We found that GMT, it will enhance PREX2's binding to its E3 ligase, HECH9. And through this interaction, PREX2 will be degraded through this proteasome ubiquitination pathway. So that means in the presence of GMT, it will enhance PREX2 degradation, and so it will lead to the activation of PTEN tumor suppressor pathway. Besides that, we found that about 23% of the Taiwanese liver cancer patients, its PREX2 gene has somatic mutation. And this is the hotspot, some amino acid mutation sites of in different patients. And all this mutation will cause the decrease of the half-life of PREX2 protein. So here we show the mechanisms of inhibition of HCC growth by GMT through this interaction with the daptol. Here we block the GMT will block the daptol's inhibition to mTOR, or through its enhancement of PREX2 degradation. So both actions were causing the AKT activations, and so the GMT played very important role in different pathways, including mTOR signaling pathway and the PTEN pathways. So besides enzymatic function, through protein-protein interaction, GMT can regulate the tumor genesis pathway. But the strongest evidence showing GMT is associated with liver cancer is this data. We generated GMT knockout mice and follow up these mice for 2 years. And we found 100% of the female knockout mice and 60% of the male mice will develop liver cancer at the age of 16 to 20 months. And he published in Hepatology and International Journal of Cancer, and we also obtained the patent. So since we show you that GMT downregulation is directly associated with the liver cancer tumorigenesis, so we think maybe we can use adeno-associated virus as a vector to drive GMT expression in the knockout mice and see whether it has some rescue effect to prevent the tumorigenesis. And this is the data. You can see that we generate AAV construct, either the GMT or EGFP, and we treat the mouse at age of 4 months old, and then we follow up for 11 months and sacrifice. Of course, every month we use the ultrasound to monitor, and also blood biochemical analysis. And you can see the results compared to the knockout mice and the control EGFP AAV construct. The mouse that received only 1 shot of the AAV gene therapy of this GMT construct, you can see very few liver nodules were found in the mouse livers. And so the number and the incidence of the tumors in the knockout mice treated with this AAV GMT construct was greatly reduced. So because of this, we decide to establish the drug screening platforms to screening either plant extracts or small molecule compounds. And this is the vector that we construct. Because we have been thoroughly studied the promoter function of GMT, so we decide to clone the upstream 1.8 kb sequence from the GMT promoter regions and link to the firefly luciferase. And we use this construct, it's a luciferase construct we put into the lentivirus. And so we generate Q7 cells stably express this GMT promoter construct. So we can add, and then we use this screening for more than 800 plant extracts and fuel compounds provided by the Taiwan Traditional Chinese Medicine Institutes. So through this drug screening program, for the primary screening, we found there are 26 hits. And then we study its cytotoxicity, and so that we limited to less cytotoxic one, so it decreased to 16 hits. Finally, we found this extracts from Peonial lactiflora pole has very high activities of inducing GMT promoters. So, and eventually we identify this compound, it is as a PGG, as an active components in these fractions. And we use PGG in either the in vitro and in vivo xenograft data. We use the 300 milligram per kilograms as a dosage to treat with mouse that has been implanted with Q7 hepatoma cells, and you can see that this is the growth curve of the tumor of the mouse treated with the PGG compared with the treated with the vehicle. So the tumor size is much smaller, that it's significantly reduced than the mouse xenografts and treated with the vehicle. And you can see that this is the PGG, the GMT in the PGG treated mouse liver, and it's being induced in the mouse liver. So this is the structure of this PGG. That's the 1, 2, 3, 4, 6 penta-O galloyl beta D glucoparasite. And this is a very interesting compound, and we can purify it to nearly 95% purities from the plants. And we found that its action to induce PGG actually is through the inhibit MYC gene expression at the transcriptional level and also in the post-translational level. It can enhance its degradation through the lysosomal pathway. Okay, so finally, I would like to report you the most exciting data is we use the same drug screening platform and screen for more than around 12,000 small molecule compounds and chemical compounds. And we identified candidates, that's the K78. And then we use the structure activity relationship analysis, and that's here. This is the K78. And then we conduct we modify the functional group of this K78 at the different functional group. And eventually we identify the K117. So this K117 has very good activity compared to the sorafenib. You can see here, this is the data that the so here, this shows that there is a dose, the cell viability of 3 hepatoma cell lines, that's Q7 cell, HepG2 and HEP3B cell. It's the growth curve were when they treated with the different concentrations of K117, you can see its survival is decreased. And in the xenograft data, we also use two dosages, 25 milligram per kilogram body weight or 10 milligram kilogram per body weight. And then we use sorafenib. This is the target therapy choice, drug of choice for the late stage, terminal stage of the liver cancer patients. So this red dot, the red curve means the sorafenib curve. And you can see that the mouse treated with the 10 milligrams per kilogram, that's a purple dot, it has quite similar effect as the sorafenib treatment. And this shows the messenger RNA level of the MYC gene expression at a different dosage of K117 treatment. And here, this is the in vivo Western blot data of the MYC expressions in the K117 at the 10 milligram per kilogram treatment in mouse liver. You can see compared to the solvent control, MYC expression was diminished in the mouse liver in different mice. But in contrast, the GMT was induced. Okay, in summary, I hope through this presentation, I can convince you that GMT is a very important tumor suppressor gene for liver cancer. Its downregulation contributes to the pathogenesis of hepatocellular carcinoma. And growing evidence, not only from my lab, but also from other labs in the world, including lab in Spain and in Italy and in United States, they also shows that GMT is a tumor suppressor for HCC, and GMT overexpression prevent tumor growth in both in vitro and in vivo. And we established a GMT promoter luciferase-based platform and use that to identify GMT inducers, both from the natural plants libraries and from the small molecule libraries. And we found this K117 compound and also PGG, this compound from the plants. And both compounds has very good activities in suppress MYC expressions, and then in return, it will activate the GMT expression. But it's worthwhile to mention that K117 only inhibit MYC expression at a transcriptional level, but PGG inhibit MYC expression not only at the transcriptional level, but also at the post-translational level through the lysosomal pathways. So the results shows that GMT-based drug screening platform is a powerful tool for discovering novel anti-HCC compounds. And the molecule, the small molecule K117, exhibits potential as a new promising therapeutic agent for HCC treatment and warrant advanced preclinical evaluation. Finally, I would like to acknowledge my colleague from Fudan Catholic University, Kaohsiung Medical University, and National Health Research Institute from Taiwan, and also Kaohsiung Veteran General Hospital and Mackay Memorial General Hospital for their collaboration in conducting all the experimentation in this talk. And my the supporting fundings is from the Kaohsiung Medical University and also from the Ministry of Science and Technology of Taiwan. Thank you for your attention.
Thank you so much, Dr. Sheng, for your wonderful presentation. I've really enjoyed the presentation myself, and I find it very informative. And you provided several gene therapy solutions against hepatic carcinoma, which I really wish they find implications very soon.
Thank you.
And actually, the audience was also very excited listening to your presentations, and we have multiple questions. So. So we can go with our first question from Brianna. She asks about the role of amino acids in cancer.
The role of what?
Amino acids.
Amino acid. The role of amino acids in cancer. I think amino acid is a building block for the protein. So its a synthesis pathway is very complicated. I mean, the metabolism pathway is very complicated. So so it's like this enzyme, the glycine aminotransferase, is add a methyl group to the glycine. So originally people did not think, associate this with its enzymatic activity or its function with cancer development. But through our studies, I forgot to mention that actually we are using messenger RNA differential display to compare the cDNA libraries between the tumor and the tumor adjacent parts of the liver cancer. So eventually we identify this GMT gene that has been only found in the tumor adjacent tissue of liver cancer patients. So I think there are a lot of important issues. I think for the presentation, the lesson we learned is one enzyme maybe it has multiple functions besides its enzyme. It can also maybe interact with other protein in different pathways through this protein-protein interaction. So as an enzyme, it can methylate transfer a methyl group to glycine, but when its enzymatic function is lost, it still can interact with like Deptol and Prex, and both protein has a very important regulatory role in mTOR and PTEN pathways. So I think this is a very good example to show that we need to keep open mind when we study the cancer biology.
Mm-hmm.
Thank you.
Yeah, that's very great. I hope this answers the question. And we also have another one. The question says, like, are there any drug-induced hepatotoxicity by using the glycine and methyltransferase?
You mean induce liver cancer? I think the carcinogens like from the environmental pollution or cigarette smoking, the benzopyrene is a carcinogen, which can induce liver cancer. And the most important carcinogen, I think, is aflatoxin B1 because it's contaminates from the fungus where it has a fermentations, and this aflatoxin B1 will also bind to the DNA, causing the DNA adults. And the most important observation is aflatoxin B1 will cause the hotspots in the p53 tumor suppressor gene. There's a hotspot, the 249 amino acid, there's the hotspot mutations that will inactivate p53 tumor suppressor, and then it will lead to tumorigenesis. So carcinogen is important. And besides that, of course, we know that that's a hepatitis virus infection also played a very important role for the tumorigenesis. Other compounds, I think the AA compound, which is also a herb, but the Chinese people sometimes they were mistaken it as a medicine, and then they use that, and then they also will cause the liver damage, and so the chronic ingestion of this herb that will causing the liver fibrosis and also kidney fibrosis, renal cell carcinomas. So I think there are many carcinogens exist in the natural environments. So we need to be very, very careful when we want to study this Chinese herb medicine. Yeah.
Yes, that's very true. Yeah, thank you, Dr. Chen, for really well explaining that. And actually, the audience are interested in knowing, like, are there any gender-specific observations that were, observed in efficacy of PGG in inhibiting liver cancer cell growth?
In terms of gender, issues. Yes, there is always a gender issues in a certain type of cancer. For example, liver cancer. Male are more susceptible to the development of liver cancer. So the ratio is about 1 to 3 to 5. So they are the higher incidence of liver cancer was observed in the male population, and there are many explanations. And my observation is actually GMT also has a gender difference in terms of expression level in the mouse liver. So male mice will not express GMT until at the age of 5 weeks old. And for the female mice, it will start to express GMT in the liver when they were born. And also the amount of GMT expressed in the female mouse liver is about 3 to 5 folds higher than the male liver cells. So you can see that there is a gender difference in terms of GMT gene expression levels. So maybe that can also explain because why there is a gender difference, because GMT it has a protective role for the liver cancer development. Besides that, people think the hormone maybe also played a role. Androgen and estrogen maybe also played a role. But of course, when people use estrogen to treat liver cancer patients, it does not have very significant effects for the tumor growth. So hormone therapy is not very effective for liver cancer. But in contrast, for the prostate cancer, hormone therapy is very important. Yeah.
Mm-hmm. Yeah. That pretty explains the issue, and also like people asking about junk food, and it's a major reason for developing hepatocarcinoma, which is obviously correct. And they were also asking about how effective targeting cancer stem cell pathways in the hepatic carcinoma, the stem cells.
Yes, for the hepatocellular carcinoma, currently the treatment of choice are still quite limited. So people would like to use like a stem cell therapy or some cellular therapy, immunotherapy for the patient at the late stage. Because for the early stage, the diagnosis tool now we can use sonogram and also some marker screening like alpha fetoprotein, so to have an early detection of a small hepatoma. For the small hepatoma, we can treat the patient with some just through the guidance of the sonogram, we can inject some liquid nitrogen or alcohol to kill the cancer cells. And so for the small hepatoma, we or even just doing some lobectomy surgery to remove the tumor. But for the late stage of hepatocellular carcinoma, now the sorafenib and the second generation of sorafenib is only the drug of choice. People try to develop some mTOR inhibitors, rapamycin, or other drugs for target therapy, but still not very successful. So I think that's why there is a met needs for identifying more potent drugs for the treatment of HCC.
Yeah, that's very true. Yeah, thank you so much. And I have a question regarding the experiment. From what I know is the use of AAV
Yes.
can sometimes be, contradicted by neutralizing antibodies. So do you normally in those experiments measure the level of the neutralizing antibodies against AAV because it might affect the gene therapy process?
Oh, you mean use the neutralizing antibody to to study the. So you're thinking of doing some infectious disease using AAV as a construct, because AAV is for the gene therapy. Usually, it's for the congenital disease. Yeah.
I actually measuring the level of the neutralizing antibodies against your treatment, because sometimes, you know, there is an immune response that can attack your treatment. So after doing the experiments, do you study this immune response, like through the level of the neutralizing antibodies or anything like that?
Well, that's a very good question. I did not do that. So maybe we should conduct the experiment to see what's the situation of the neutralizing antibody to the treatment. Yeah, thank you for your suggestion. Yeah.
Yeah, you're very welcome. And thank you so much for your interesting presentation. We're really glad to have you today.
Thank you.
We're looking forward to collaborate more in the future. Thank you so much, Dr.
Thank you very much. Thank you.
Thank you.